Your zip code may be more important to your health than your genetic code. We used to think chronic diseases like heart disease, diabetes, some cancers were really a product of the genes you inherited and the lifestyle choices you made as you were getting older. What epigenetics tells us is that these diseases, rather than starting later in life, actually occur at the earliest stages of development and probably even in the previous generation. People are being born into this world at fundamental disadvantage that actually increases over time as a result of interaction with hostile environments. It's not biological determinism. It's about risk. And what we know is that risk that somebody comes into the world with is either reduced or increased by the environments they come in contact with.
Quantitative genetics ties back to Mendelian genetics in that we're looking at phenotypic variation. However, in quantitative genetics, we're interested in what we call continuous phenotypic variation. This is found in traits that show variation in a continuous range of phenotypes rather than discrete. So in Mendelian genetics, we looked at discrete traits like dwarfism versus tall, yellow versus green seed pods. Here we're looking at this continuous range. So for example, height in humans. Right? We're not either tall or dwarf. There's a whole myriad of heights. Or say milk and meat production in animals. Yield in seed protein. All of these things are continuous traits. And continuous traits follow what we call quantitative inheritance, meaning that continuous variation across a range of phenotypes can be measured and described in a quantitative term, so in terms of discreteness to the concept of polygenic traits. So we talked about quantitative traits being traits that have a continuous variation, and we can usually describe and measure these traits based off those quantitative terms. We also have what's known as polygenic traits, in which the phenotypes they're being result from both input of many genes. Okay, and then we have multifactorial traits that, again, can be a reflection of a polygenic trait or a polygenic gene in which both that gene action is giving the phenotype as well as environmental influences. So if both, a straight polygenic trait is just the result of genes, a multifactorial is the result of genes and the environment. Furthermore, we can kind of divide polygenic traits that don't show, say, a continuous variation into what we call meristic and threshold traits. So a meristic trait is where the phenotype can be recorded by counting whole numbers. So this would be like number of seeds in a pod, right? We don't get 2.4 seeds or 3.2 seeds. No, we get whole numbers. You either have 2 seeds, 3 seeds, and so on. Or the number of eggs laid by a chicken. That's a meristic trait. Okay, threshold traits are polygenic and often multifactorial. And in this case, what that means is that you have to have a certain number of alleles before we start expressing that phenotype. So an example of this would be like type 2 diabetes. This is the adult onset diabetes. You could have a lot of predisposing alleles in your genotype for diabetes, but it's not until you cross this threshold zone that we'll actually have diabetes. Now, when we talk about these if we look at examples of these quantitative traits, what we see is that in these quantitative patterns that there is still Mendelian factors at play, but they can't account for the range of phenotypes seen. So looking at this wheat shaft, if we have both red and white, we'd expect to get this intermediate color. And then we get all of these additive alleles. That then when we self-cross the intermediate color, we end up with these different variations of color. And that can't be predicted just by Mendelian factors alone. And so in traits where we see this, we developed what's called the multiple gene hypothesis. And it says that many genes individually behaving in a Mendelian fashion can contribute to the phenotype in a qualitative or quantitative way. So what that means is, while this right here obviously follows Mendelian genetics, we have this intermediate, we then break this down and say, okay, if we self-cross these intermediates, these F1s, we're going to get all of these different genotypes. But each one of these genes functions individually towards an expression of color. And so we actually have 4 additive alleles, so 1, 2, 3, 4. And if you have all 4, then you're going to be the dark red. If you just have 3, you're going to be this dark medium red, right? So we can see down here, here's the colors, 4. And when we have 0 additive alleles, that would be in this case, the additive allele is going to be the dominant allele. We would just be white. Now, in this multiple gene hypothesis, we're assuming that we have both additive and non-additive alleles. So in that previous example, the additive allele was the dominant allele. The non-additive was the recessive. All additive alleles contribute equally, in that previous example, to red grain color. The non-additive alleles fail to produce any red pigment. And so the greater number of additive alleles in a genotype, the more intense, in the previous example, the more intense red color is expressed. So this is this concept of quantitative loci, that we have these loci within our genome that function in a very similar fashion and that they control various quantitative phenotypes. And the more we have of those quantitative loci, then the more complex our ratios are going to become. So let's just look at this. If we had one gene pair, we would expect 3, well, really just 2 outcomes, right? But it's going to be 3 phenotypes. If we have 2 gene pairs, we're expecting 5 different genotypes. 3, we're getting up even now to 7. 4, and you can see how it goes higher and higher, and we're getting these different ratios. Additive alleles are the basis of continuous variation in quantitative genetics. We say, okay, the more these individual alleles contribute to a single quantitative character, the more those will add up, and the phenotype will be a contribution of each additive allele, and not one allele is not greater than the other. They're all equal. So as we take our additive alleles and we put them together, we produce these substantial. Now, if we want to identify the number of polygenes present in a particular trait, we can do that by looking at the F2 generation, and we want to see the number of individuals that resemble either one of our 2 extreme parental phenotypes. So really we're looking for the ones that are either all dominant or all recessive. And then the equation we would use is 1 divided by 4 to the nth power. The n is going to be the number of polygenes. That should equal the ratio of F2 individuals expressing either extreme phenotype. So for instance, if we had a F2 and 1/4 of the offspring represented one of the extreme phenotypes, then the number of polygenes would be 1. Why? Because 1 divided by 4 to the nth power, which would be 1 in this case for 1 polygene, is going to equal the ratio of F2. So it would equal 1/4. If we actually had 1/16, then we'd have to put a 2 in there. Okay, so we can solve for this. There's some equations you can go through to utilize this to solve for this, or you can just type in these numbers until you get the right number that works, right? So let's do an example. It says in a certain plant, height varies from 6 to 36 centimeters. When a 6 centimeter plant is crossed with a 36 centimeter plant, all F1 plants were 21 centimeters. In the F2 generation, a continuous range of heights was observed. Most were around 21 centimeters, and 3 of the 200 were as short as the 6 centimeter P1 plant. Okay, so what mode of inheritance does this illustrate, and how many gene pairs are involved? Well, we have this continuous variation, so this would represent a quantitative trait. It's polygenic. Okay, and if we want to solve for how many gene pairs are involved, we would do that by going, okay, what was the ratio of extreme phenotypes? 3 out of 200. So I take 3 divided by 200 is going to equal 1/4 to the n power. Okay, now when I break this down, 3 200ths is equal to 1/64. So that's going to then equal my 1/4 to the n. And now, the easiest way I would say to solve this without getting, I mean, you can use algebra to count for n, but if you don't remember all that, I just start plugging things in. So we know 4 to the 1 is going to be 4, 4 to the 2 is going to be 24, 4 to the 3 is going to be 64. So n is equal to 3. 3 polygenes are involved. Okay, so now for this next question, how much does each additive allele contribute to height? Well, how do I solve that? Well, we know if I drew out our height, we know that 6 is at the bottom, 36 is the most. So I can solve this by taking 36 minus 6. So I take my two extremes, subtract them from each other. That equals 30. So 30 centimeters divided by the number of alleles. How many alleles do I have? Well, I have 3 polygenes, so that means I must have 6 alleles. So if I take 30 divided by 6, that's going to equal 5 centimeters per allele. Everybody follow that? So the last one says, list all genotypes that give rise to a 31 centimeter plant. So if this has 0, this has 6. So 6 plus 5, my next one is going to be 11 centimeters. These ones should have 1 additive allele. Add 5, then I should get 16. Add 5, then I should get 21. This should be 2, 3. Add 5, I should get 26. 4. Add 5, I should get 31. It's 5. Add 5, and I should get 36. So list all the genotypes that give rise to a 31 centimeter plant. Well, that's going to be all of the genotypes that have 5 of additive alleles. So if we want to answer this, again, we have 3 traits. So AA, that's 2. B, okay, that's 4. And then I do a big C, little c. right? That's one genotype. It could be a big B, little b. That'd be two genotypes. It'd be a big A, little a. That would be three genotypes, right? Or right? Those are the genotypes. Are there any other ones? You guys think about that. So when we study polygenic traits, we have to rely on statistical analysis because we're looking at populations, and we have to assume that those frequencies of what we see in the population would fit a representative, what we call normal distribution. So we measure lots of large samples and we look for representative individuals in that population. We create, a normal distribution curve. It's like a bell curve, and we would plot a frequency histogram. If we are talking about the actual heritability of a trait, so heritability values as an estimate of the genetic contribution to the phenotypic variation, we are now taking into an assumption that, hey, we're looking at multifactorial traits in which genetics make up a part, contribute to it, but the environment may as well. So heritability describes the proportion of total phenotypic variation in a population due to just genetic factors. It does not indicate how much of a trait is genetically determined or the extent to which an individual's phenotype is due to that genotype. It's just looking at what's the relative proportion that's probably due to genetic factors. This can then allow us to what we call a heritability estimate. This gives the proportion of a phenotype, what proportion of that phenotypic variation can be attributed to genetic variation within a certain population in a particular environment. This is going to change if populations have different environments, right? So if we had a heritability mean of 0.65 in human height, what does that say? Well, it's telling us that 65% of the overall variation that we see in height is due to genotypic differences in individuals. Okay? That's what it says. It's not the same as saying 65% of all height is due to genes. No, that's an oversimplification. We're saying 65% of the variation in height can be explained by genetics. We can take this variation and we can start to describe then what we have. We have phenotypic variation. That would be all variation that we see in a population on a phenotype. That would be V sub P. Okay, and there's different components that make up that phenotypic variance. One is what we already talked about, the genotypic variance. Okay, we also have environmental variance. And then we can have interactions. We can have genotype by environment interaction variance. So if we look at in this particular example, we're looking at varieties of corn in different soil fertilities. So we can see in a low soil fertility versus high soil fertility, we get different yields between these two varieties. Right? That would be where we have no genotypic environmental interaction. Okay, so here's the environment basically right here. So as the environment changes, we see pretty much the same yields, like we have this increase. If, however, there is an environmental and genotypic interaction, we might see something like this, in which variety A stays on this course, but variety B has a dramatically different slope. And that tells us that, hey, it's not just the environment, there's also some genetic interaction going on at the same time, so we have to look at that interaction. And we get these heritability estimates using experimental and statistical techniques, right? So we can partition all of our phenotypic variants into what we call genotypic variance, which is V sub G, and environmental variance, which is V sub E. And from that, we can calculate a broad sense heritability. So broad sense heritability measures the contribution of genotypic variance to the total phenotypic variance. It's designated capital H squared, right? So we take our V sub G, the variance of genotype, divided by the total variance we see in a population. So broad sense heritability estimates, they're assuming that genotype by environmental variance component is negligible. We're not bringing that in when we do a broad sense. On the opposite side, we can do what we call a narrow sense heritability. This is the proportion of phenotypic variance due to additive genotypic variance alone. And you might say, what is that additive? We want to know what's in the narrow sense heritability, what proportion of that genetic variance is due to just additive alleles. And in order for us to do that, we have to break apart what makes up genotypic variance. There's 3 subcomponents of it. We have that additive variance. So this is the variance from additive alleles. We have the dominance variance. So this is the amount of dominance that is playing a role in that particular phenotype expression. And then we can also have an interactive variance. Okay, interactive between the environment and our genetics. So just to recap, additive variance is that genotypic variance due to the additive alleles of a quantitative trait. Dominant variance is the deviation from the additive components that result when phenotypic expression in heterozygote is not precise because dominance is overplaying some of those additive alleles. And finally, that interactive variance is the deviation from additive components that occurs when 2 or more loci behave epistatically. Right? So we might not know this. Here's a table from your textbook and where they look at different traits in different organisms, and they've calculated out the narrow sense heritability. So you see like tail length in mice, 60% of that variance found in the phenotype can be explained by just additive alleles. So let's do a problem. It says a hypothetical study investigated the vitamin A content and cholesterol content of eggs from a large population of chickens. The variance, V, were calculated as shown here. So we have our total phenotypic variance, our environmental variance, our additive variance, and our dominant variance. It says calculate the narrow sense heritability for both traits. Which trait, if either, is likely to respond to selection? So where do I start on this? Well, I need to remember that my narrow sense heritability is equal to the variance of additive alleles divided by variance environmental plus variance additive plus variance that is dominant. Okay, so I can go through and now type these in. So I can take for vitamin A 12, I'm going to take 12 divided by 96.2 plus 12 plus 15.3. And that's going to give me 0.097. Okay, for my cholesterol, I'm going to take 192, 1, and divide it by, again, 484, 6 plus 192.1 plus 185.3. is equal to 0.223. So which trait is more likely? Well, if I multiply that, this is equal to basically 9.7%, and this is equal to 22.3%. That's our heritability for both traits. So which trait, if either, is likely to respond to selection? Well, it's probably going to be the cholesterol, because 22.3% of that variation can be explained by additive alleles. And so that's going to get us a lot further if we're selecting chickens that obviously have whatever we're going for, say low cholesterol on their eggs. We could probably see more of an effect through selective breeding for that than we would for vitamin A. Now, a lot of studies have looked at trying to decide which, phenotypic traits or which quantitative traits in humans, what is the broad sense, narrow sense heritability for some of these traits, and we find using humans twins incredibly useful. Why? Because the phenotypic variance for a multifactorial trait due to genotype as opposed to the environment can be explained in human twins. So if we have a monozygotic twin, that means identical twins, they're derived from the same egg, single zygote, we would expect that phenotypic variance is going to equal the environmental variance, because there shouldn't be any genotypic variance between those two individuals. They have the exact same genes, loci, alleles. Whereas if we look at dizygotic, that means fraternal twins, they're derived from two separate fertilization events. The phenotypic variances are going to represent environmental variance and approximately half of the genotypic variance. So we can start to say some things about what's going on as far as broad sense heritability. Again, broad sense heritability estimates, they're going to estimate and provide a comparison of phenotypic variances for same traits in identical and fraternal twin sets. And we can talk about this concept of concordance. So when twins are concordant for a different trait, what that means is that they either both or neither express that trait, right? If they're concordant. If they're discordant, that means that one will express it when the other does not. And we can compare those concordant values of monozygotic versus dizygotic to give us some information about what's the potential value for heritability of these traits. Now, we are always limited in twin studies by size. By the time they're born, most monozygotic twins don't, they don't have necessarily identical genomes. There are some slight changes that occur. Additionally, we have gene expression patterns in monozygotic twins that change with age. So at different ages, we could have phenotypic differences. Looking at, this is percent concordance between monozygotic and dizygotic, we can start to assess, what's the relative importance of genetics in a broad sense heritability standpoint versus environmental factors for each of these traits. So we can see, like blood type, 100% concordance. That tells us there's a high broad sense heritability for this trait. Even in dizygotic, it's 66. That's still pretty high. As we start coming down, though, we start to see some of these things change a little bit. So like hair color, high concordance here, but it's not as concordant in the dizygotic. So while broad sense heritability makes up a huge role, there may be a small contributing factor of the environment. However, some of these other ones, right, are environmentally driven. Clubbed foot or mammary cancer seems to have while there might be some broad sense heritability, it's much more likely that it's the environment. So that's the end of this chapter. Make sure that you go back and you look at the study guide, you look at the learning outcomes. You're going to need to be able to calculate broad sense, narrow sense heritability and make some decision points off that. You're going to be able to have to calculate the number of polygenes present in alleles. So make sure you practice those problems.